Hi everyone, I'm Maggie. I'm going to be walking you through the split pea DNA extraction experiment today. But before we start, I'm going to talk a little bit about DNA and the role it has in living organisms. So first, what is DNA? DNA is the molecule that's made up of two polynucleotide chains, and it provides a mechanism for passing genetic information from one generation to the next. The role it plays in living organisms is DNA is responsible for the encoding information that organisms use to reproduce, grow, and develop. Okay, let's get started on the experiment. Split peas and our 2 containers of salt. First one. To that, we are going to add 1 cup of cold water. And we're going to blend this for about 30 seconds. Blending, we're going to take our lid off. We have our cup ready here with our cheesecloth and rubber band. We're going to start off by putting on our safety glasses. We're going to take our cheesecloth, put it right on top of the cup here, and we're going to secure it with this rubber band. It's okay if you need to move it a little bit. Okay, our next step in our through the cheesecloth. And now we're going to remove all of this content here and discard of it. Now we have our strained fluid in this drinking glass. We're going to take this dish soap, put all of this in here, stirring with our glass rod. And we're going to put this in the fridge for about 10 minutes. This test tube, and we're going to pour this in here and away here. Now we are going to take this meat tenderizer, and we're going to pour about half. We're going to take our glass stirring rod, get this stuff done. Now we're going to get our chilled alcohol out of the freezer. We have our 91% here. Next is hold this test tube at about a 45-degree angle and fill up about another third with, here we are at an angle. And here we are with our alcohol. Now we have this in an upright position. We're going to allow this to settle for just 1. While we're waiting for this to settle, we're going to go over some of the questions that we have that go along with this lab. One of them is, why do we add the sodium chloride here? We add the sodium chloride because it binds to the negatively charged DNA, and this neutralizes what we have going on in here. And we also, the DNA clump together. And then why do we add the detergent? We need to physically grind the material to disrupt the cell wall. And the detergent causes the cells to open, causing the DNA to be released. Why do we add the meat tenderizer? We add the meat tenderizer to both separate and isolate the DNA from the surrounding proteins left. Our next question is, what is the purpose of the chilled alcohol? And the chilled alcohol increases the amount of DNA produced, and it speeds this whole process up. And the temperature also affects the solubility. So as the temperature will naturally decrease as this alcohol chills off, so will the solubility of the DNA as well. And now it's been about a minute, and we can see that we have some clear separation here. And I want to take a minute to make some observations. So here it is up nice and close. We can see that we have some white stringy DNA strands going on all in here. And it looks like we have some good quality DNA. I mean, that's a good chunk of this test tube that has our DNA in it. And it honestly, it looks pretty thick too. And we have our extra of this pea-colored water under here. Our chilled alcohol we can feel is up here toward the top. And then our DNA is right here in the center. So thank you for following along with this experiment.